Hi, sometimes tutorials are like a menu in the restaurant. You look, you read, and you still don't know what to choose. But you always can try first and then decide whether it was good for you or not. Previously I told you a story how to use exact test in edgeR protocol. You notice that exact test is only applicable for experiments with a single factor, which is most of the time not the case. And further, right after this, authors say that if it is a more general story with many factors, then you use generalized linear models. And before that, you estimate dispersion, which is based on Cox rate likelihood method. And to do so, you write this line, or it can be two other options written here. But then the question is, and what then? Then you write lines like this and you use a glmFit, which is probably the last option in a variety of edgeR protocols. The major lines are somewhat similar to this classic storyline with the single factor, only these two lines will be replaced. You do glmFit according to design, and then you select the contrasts the way you like it, and find your differentially expressed genes, and here it will be this value for a top tax, and then you go to a tops, etc., etc. I call this protocol alternative glm edgeR. Why alternative? In a chapter 3 you will see the glm approach is reserved for the script with the glmQLFit option. Those functions glmFit and glmQLFit sounds pretty close, and you might think they do something similar, but they're probably based on different algorithms, and you are quite right with this. QL abbreviation comes from quasi-likelihood methods, and the glmQLFit function is more integral compared to glmFit. It does not require a separate line of a dispersion analysis. As usual, I decided to expand those lines and make a complete working script. For this, I have chosen another data set because I'm still doing my mini study on aging in hematopoietic stem cells in human, and the previous attempt failed. There were no differentially expressed genes found, so I tried this data set. So this is one more set of aging hematopoietic stem cells. There is a publication, and there are a reasonable number of samples, young and old, better balanced than before. And as usual, I downloaded not normalized counts but raw data. And after this, I got a collection of the files. So one sample in one file, and therefore I used the same readdge function to make a data table. So now it is a data table with a pretty long names. There is no age, gender, or any details in it, only a sample and a sequencing number. Therefore, it had to be further processed. Yeah, there are NAs in the data which has to be removed. Now I used the old trick. I import sample data with a getGeo function out of the site. As usual, it has lots and lots of information which you don't use. You will need this age and sex for sure. some other columns may be. I decided to use this for and call the table reduced. So now we have a young and old and male female. There is again a problem with that female are prevalent in old samples and the males in young samples. So the gender bias still exists in this data as well. Alright, let's continue. Now we have to process sample table anyway. Make alike row names in reduce to the call names of a data. You have to be careful with this operation. Make sure that indexes of this sequencing data are the same. They are all sorted in the right order, so there is no mistake. Then you generate young and old group, because again ages are not converted into groups. There are over here, so young and old should be extracted, or this should be used, which was not used. But this column is also not usable immediately because of this. Alright, we continue. Simplifying the sample table. And this is the final table that the group of young and old are formed. The sex is also here. All the rest is a supplementary information. Row names are exactly the same as the column names for data. It is important, so we're ready to go. If you do it for yourself, you can save the table, not to repeat it next time again. Have a shortcut. Then we declare edgeR list. Right after edgeR list, you check samples. It's all agrees. Now you could think about box plot, but wait a bit, because you better filter data first, and then before normalization you check the box plot. It looks okay, they look comparable, and then we do normalization and we check again genes and reads as a plot. No outliers detected. This might be a little bit high in read numbers, but in gene numbers it's okay in the middle. Therefore it will be well normalized and adjusted. Then we do MDS. If you think gender makes some samples to stand out of the clouds, it is not. Number 19, for instance, is a female, just as many others in old group. And 14 is a male, which is not standing out in old group, and so on. There is no problem with the gender outliers detected. So then we make a design group and sex together, and then we estimate dispersion, not a common, but just dispersion. And remember, you can estimate dispersion in one line or in two lines, like this or like that. You can also visualize it. Note that we have no visible outliers, and now the dispersion cloud is pretty compact. That's how it should look like in a good case scenario, that there are not too many dots flying out of the major cloud. And even more, if you check the y variable, what is new after dispersion estimation, that you see that there is a common dispersion of 7, that's the red line, and then trended and tag-wise, which again is a bit difficult to grasp, but it's simply try to follow the clouds. So more detailed dispersion function. It gives the coefficients for every average log CPM value. That's what is needed down the lines. Alright, and now we come to glmFit, because this was the major news in our protocol. That's what makes a protocol different. And then you also check the coefficients. I followed the model without zero, which I hate, but that's what authors recommend. And then you call the results based on coefficient 2, meaning that group young is compared to the rest of the data. That's the difference between explicitly calling columns for comparison using contrast matrix. In this case, you add 0 to the model and exclude intercept, or you do it the way how author suggests. But there is a difference. It's not explicitly compared to other second group. It compares to everything what is not in group young. To check whether it matters or not, I added 0 to design and repeated the script from here. Obviously dispersion is the same, but then after glmFit, which is still the same, you ask for design and the columns are different now. And now I insert a make contrasts line where I explicitly say compare me these two groups with one another. Then I run glmLRT, and then I check how many tops I get with exactly the same conditions, and it is exactly the same number, 748. Note that this dataset has only two groups, and for two groups it seems it makes no difference how you write the model. Let's continue with original design. We do next mdplot, which is a replacement of volcano plot. Somehow, if you look into the edgeR tutorial, they also prefer this plot to the volcano plot. And then we come to a top tags. It's all meaningful. Always check that you did it correct, so there are actual numbers behind p-values and FDR. If zeros are here, then it means something went wrong. And then you generate TT table, and then you reduce TT table to the tops only with FDR less than 5%. You check the size, it's 748. But there is a problem, it's not yet a names over here. Again, I use the same trick. I call the library, it's a human data, so I call a human annotation. I check the columns. In this particular case, it's not entries, IDs, but ensemble numbers. Before that, you have to remove these suffixes, these numbers after the dot and dot itself. You do it by gsub function. You can check Stack Overflow. There is a discussion about it and the recommended lines. So you copy paste and do it like this in two steps. First I do ensemble keys, then I check the keys. Now no dots, no values after the dot. Then I have the original table, and I verify that the order stays the same. Usually from first line to the last line, the order of the row names is the same. And then you do a selection of annotation. One to many, it means that there are more lines with the same gene name. There are some multiple transcripts associated to some genes. And then you merge those two, and then omit those which have no gene names. And this is your end result, which you can enjoy and do something else. This was the last edgeR protocol, which I could find in the edgeR tutorial. And if you still don't know what to choose, take an advice from the kitchen. Use glmQLFit or glmFit. They are of rather general use to a wide spectrum of the data.